So, unfortunately, because of the timing of the exam and everything, I won't do a review this time. So I would recommend that you get with study buddies and review. This is a pretty technical exam. Do you have any questions about the exam? Yeah. Yeah, it's about 90. Right now it's sitting at 98, so I'm going to probably cut a few, but yeah, it's sitting at 98. So we'll see. Yeah, so kind of like last time, you're going to have metrics. Don't miss a metric stuff, right? If you're missing metric stuff, get it fixed now, okay? Please, please, please. My night class, we stayed until 7 o'clock writing on the board, doing metrics problems. Okay, just practicing, okay? There's really, don't miss those questions. Don't miss scientific notation questions, please. Those are the easy things. And if it's not easy, let's work to make it easier, okay? Yeah, so that's about it. How many are working on your note sheets, right? Okay, yeah, I probably will have some extra student hours this week because of the exam, so you can come in and ask questions. I just have to see if I can get out of one meeting, and then I can get more student hours. So that's all. Yeah, anything else on your mind? The quiz average went up, so the quizzes averages are going up. We had some 9s and 8s, and that's great. Okay, so let's talk about the genetic code here. Okay, wait a minute. Where's my thing? Okay, this should be kind of a review about genetic code. We didn't do this last time, did we? Nah, okay, so let's talk about the genetic code very quickly. Are you going to have to memorize the genetic code? No. I'll give it to you. Do you have to know how to use it?
Yes.
Yes, okay, good. Wow, everything is disappearing. What is that? Oh, there we go. Okay. So, like baseball, it's a baseball time of year. So we go from first base to second base to third base. So let's give you a couple of things to see if you can do this. I'm going to write a sequence up here on the board, okay? And I want you to tell me what amino acids it codes for. Okay. So how many codons do I have up here? 4. So how many amino acids should we have? 4. Good. So just do it right now. Try and do it because you needed to read before you came in, and you've had this in Bio 1 and you've had this in anatomy. So yeah. Okay, you got it? Almost, okay. Okay, what'd you get for the first one?
Start code.
Okay, that's a start codon, but it codes for an amino acid. What does it code for? Methionine, right, so met. Okay, CGC, what did you get? Arg, the pirate one. Okay, it was arginine, okay. UUU. Phe, which is phenylalanine. And then what is this one? Stop. Does this one code for an amino acid?
No.
No, okay. I'm going to ask you to do some sequence like just what we did on the exam, okay? If I see start here, it's wrong, okay? Because yes, it is a start codon, but there is an amino acid that goes in that place, okay? Is that clear? Don't miss that. Okay, that's easy sneezy. So, okay, so we've got the chart. You can see some redundancy in this chart. Okay, like it's usually the first 2 bases of the codon that you can see, and then you've got a little wobble here. Especially like look, you can have a little wobble here. Serine has a ton of wobble, and it's called the wobble hypothesis because those first 2 line up really well between the codon and the anticodon. But that third one, it can wobble a little bit. Okay, good. Okay, so let me switch gears here and talk about translation. Okay. Got a question? All right. Oh, don't want to auto load. Oh, maybe we do. Let's see. Okay. So, gonna give you. Wow, this is from a long time ago. We learned a lot. Okay. Okay. We talked about post-translational modification. All right. So, I want to give you 1 minute to explain translation to the buddy sitting next to you. Okay. 1 minute. All right. We start with 1 minute of translation. Okay. Make sure to draw what's going on.
And then the amino acid range of these, and then it starts dividing. You can't read yeah, and it continues to go until the stop codon, which it then releases the
I think so. I think oh yeah. I need turns to yours.
Okay, let's talk about translation. Please do not confuse transcription and translation. Okay, that's a really common thing to do. Get it on different papers, do something so you can remember that these things are transcription, these things are translation. Okay, so what do you need for translation? Let's start with messenger RNA. Okay, who comes in first? No, we're way past that. This is translation. Yeah, way past that. Who comes in first? Okay, which part of the ribosome? The small subunit. And what is it looking for? Yep, so the small subunit is going to scan along here and it's going to stop when it sees AUG. And it will encompass 2 codons. Okay, it encompasses 2 codons. All right, now, who comes in next? The large ribosomal subunit. And what are the sections on the large ribosomal subunit? P, A, and the exit site, E. Okay, who's coming in next? tRNA. tRNA will come in. Okay, and it will have an amino acid. In this case, it's methionine. Okay, ta-da! And this region of the tRNA is called the what? The anticodon. That's going to bind to the codon. And that's set. And once we've got this all assembled, okay, we can start. Now, what happens next? Another tRNA comes into the A site. Okay, ta-da! Okay, and the tRNA binds to, the anticodon binds to the codon. Okay, and we've got CCC, which I don't remember which one it is. I think it's phenylalanine. But, okay, now what happens? Nope, not yet. This is where the magic happens. Yeah, I know what you're saying. What kind of bond is made here? A peptide bond, right? It's a peptide bond that's made there. Okay? Once that is done, this tRNA right here, as soon as it transfers that amino acid, this is considered what they call uncharged. It doesn't have an amino acid. So where is it going to move to? The exit site, right? It's going to move to the exit site. Okay, that's lovely. So let's get a tiny eraser and do that. Oh, that's a tiny one. Okay, all right. So this amino acid moves over to the exit site. And what happens to, what happens to this whole complex? What is it going to do? Yeah, it's going to translocate. So now this is in the A site, and now you can have another amino acid come to the P site. Or the, yeah. So this is P, this is A site. Okay. Okay. All right. So it's clear. All right. This is P. This is A. Okay. Now what's going to happen next? Yep. Going to make that peptide bond. Ding. And then what happens? This tRNA is now uncharged, so it's going to move to the exit site. And what's going to happen? It's going to translocate until it hits the stop, UGA. Wow. Yeah. That's one of the stop codons. There's 3. Okay, what happens when it hits the stop codon? What happens when it hits the stop codon? What? Releases. Okay? This polypeptide gets released. Okay? Everything falls apart. Okay? The small subunit, the large subunit, everything falls apart. Okay? Can this mRNA be used again? Absolutely. Yeah. Okay. Good so far? All right. Okay, I'm going to turn on the lights. Now explain this to whoever didn't explain last time. Explain it to your partner, how this works. Okay? Oops. Come on. There we go.
The tRNA will restart. They're up together and they translate. And then we're going into this, right? The start codon goes through a little exit house, right? The start has the PUG. It goes, oh, yeah, it goes to the
They separate and then the polypeptide
it's not those 2 bonds, you're like, no.
Yeah.
The next sequence, maybe. So it's like, I'm not ready for the 2. We're not ready for the 2.
I didn't get a call.
No, that's what that was the way. That's a big too.
Okay, what are your questions after you've had to draw it from memory? What are your questions that you have? Yeah.
The mRNA disintegrates once it's released, right? Or what happens to it?
Well, it will stick around for a while, and in eukaryotes, the message eventually will be degraded because it's got, once that poly-A tail is gone. Usually that's the side that the braid's on, not always, but it will stick around. It will stick around for a while, but there are RNases around in the bacteria and in eukaryotic cells, so that message may stay around for a little while, get transcribed a few times, and then it will disintegrate. Yeah, good question. What else? What else are your questions? Okay. This is one of those things that you got to be able to draw from memory, okay? That you got to know what comes in when. And one of the common things I saw as I was walking around is that people were like, okay, so here's your mRNA, and they were trying to put the ribosome just over one codon. Doesn't work, right? How many codons should it be over? 2. Yeah, good. I saw a hand go up. Question?
No, you just.
Okay, yeah. Yeah, you can't do this, right? No. Because why? It's built to do this. So you have codon 1, codon 2, and that small ribosomal subunit comes in and encompasses 2 of them. So adjust your drawings, because you're going to get home and say, well, I don't get it. Yeah?
Did you say that tRNA is empty from the P site?
Yeah, so once it transfers that amino acid, it's uncharged. And then it goes on its way out. It goes to the E site and then goes into the cell and hopefully picks up some amino acids. Yeah. And recharges. Good question. Yeah.
So it uses that codon right there, and then once it's done with it, it moves to the next one. It doesn't use the CCC for this
No,
one.
it's going to use here, and then it will translocate to the next one.
And it won't even, that one's done with.
Well, this is the P, this is the A. First it moves, so now this is in the P site, this is in the A site. Then it moves, so this is in the P and this is in the A site. You
Okay.
still look confused.
So it does use the CCC, right?
Oh yeah. So that's going to be here, that's going to be here, right?
Yeah.
Okay, it transfers, this goes away. Whoops, sorry, I didn't mean to erase the amino acid there, that's not good. Okay, and then where is it going to move? It's going to now encompass this, right? And now this becomes the P site, this becomes the A site.
Gotcha.
Okay, cool. What other questions do you have on this? Good? All right. Okay, where does this happen in the cell? Cytoplasm for both prokaryotes and eukaryotes, right? Good, okay. Does it help to draw through these and work it out? Okay. I know it slows us down a little bit, but that's all right. Okay, there's all the words about it, blah, blah, blah. Yeah, see? Look at that. This one's upside down. That's okay. They can do it upside down or right side up, right? So there's that. Then moves over. Okay, then it moves over. Then it moves over, and then the stop codon, it does not have an amino acid in there. It has a release factor. And then everything falls apart. Okay? All right, I'm going to ask you a thinking question now about all of this. What happens when the tRNA cannot get an amino acid that it needs? Yeah, what happens? It's what? It's stuck. Okay. So can it make a protein if it can't get all of the amino acids? No. Do you see why childhood malnutrition is so bad? Because, I mean, you go to countries where, you know, there's significant malnutrition in children, and what do you notice about their physiology? They're smaller. They're smaller. Their bones aren't as strong. Their immune system isn't as strong. If you're not getting amino acids, can you make all your proteins? No. Will it have effects on your phenotype? Absolutely, it does. And can you reverse that just by saying, okay, well, you know, now you're 6, I've got some more, you know, protein. Okay. So it's the same with bacteria. some of the chemicals that we're going to look at now, because you might be asking, why in the heck are we doing such detail about protein synthesis? Because we're going to do chemotherapy, antimicrobial, any chemicals to control microbial growth coming up pretty quick. It's chapter 13. And a lot of them affect this. A lot of antibiotics and antimicrobials affect this process here. This is the most energetically expensive process that cells can do, is making proteins. That's why they don't want to make them if they don't have to. You ever wonder why when you're fighting an infection you're so tired? Well, there's inflammation and there's tissue damage, but you're also trying to make antibodies. And what are antibodies made of? Proteins. Yeah. So if it costs a lot of energy to make proteins like this. Okay? So anyway, so eat your protein. Okay. Any last questions about this before we go on? Okay. So there's some activities in the PowerPoint you can do about what direction things are. This is a review about protein structure. The structure is related to the function. You should remember this. This was chapter 2. Okay? Go through and see what kind of bonding is important. See what kind of folding is important. What shapes are important. Okay? All right? So let's talk about diseases that have misfolded proteins, and that is prion disease. Okay? Folding, the structure is so important. So here's prion disease. There was a professor that discovered prion disease at UC Riverside, actually, is where they discovered it. And it's not a virus, it's a protein. It's a misshapen protein that goes and changes the shape of other proteins. Okay? So it misfolds, okay, and then it causes other proteins to misfold. So these are 3 prion diseases that we see. Kuru, which we see in Papua New Guinea, and groups of people that are cannibalistic, okay? In this particular region, when somebody dies, what they do is they make a soup of the brain and spinal cord, which is where these prions hang out, for the women and children. And so you see kuru in the women and children. You don't see it in men particularly, unless they're eating a lot of brain, okay? I ate brain. I thought I was eating a mushroom, actually. It turned out to be brain. It was quite tasty. Yeah. I was at a friend's house, and they were having a lot of Middle Eastern food, and I was like, man, these mushrooms are awesome. And they're like, Heather, it's not mushrooms. It's brain. Yes. No. Oh, no. What kind of party do you go to? No, I think it was sheep. Sheep brain. Yeah.
It's bad if you eat a lot.
Well, it has to be made sure that it came from a healthy animal. Yeah, it has to come from a healthy animal. So, we saw some really advanced Kuru and classic CJD in soldiers that were in the Middle East in the first Gulf War. They were told, don't eat from street vendors. You cannot eat from street vendors. And what did they do? They ate from street vendors. And for some reason, they got this really aggressive Kuru. I mean, usually it takes about 20 years for this disease to develop, but they were dead within 5. They were treated in Walter Reed, and there's really no treatment for this. Once you get it, that's it.
5 years?
5 years they died. Yeah. So that's why it's like, we don't talk about this disease a lot, but it's one of those things. So a lot of people don't hunt in California, but where I grew up, we got off school days to go hunting, right? And oh yeah, because we needed the meat for the winter, right? So yeah, so we had freezers and stuff, and we have, you use the whole animal. But when the animals started showing up with scrappy, like the deer and stuff, and they were acting weird, you don't eat those because they may have a prion disease. So back then we didn't have to worry about it. Now we do. So yeah. So any questions about prion disease and protein folding? Yeah.
Okay, so they don't sell that here though, right? Brains?
Oh yeah. Oh yeah. Yeah.
I've had it, but not in this country.
Yeah, I've had it here. Yeah, I've had it here. This wasn't I ate capybara in other countries, but I ate brain here. In Chatsworth, actually. Yeah. Yeah, it's very common in the Middle East, and it's actually very tasty.
Yeah.
Yeah, but I didn't know. I thought it was mushrooms. What a dork. Anyway. Okay. Wow, I didn't know that topic was going to be so exciting. All right. Yeah. No, it's actually really, really, yeah, it was delicious. What? I haven't had that. It's pretty good? Okay. There's lots of taste to have in this food. I love it. Okay, so you're kind of getting there. Here's a preview into where we're going. We're going to look at interference with RNA polymerase. What process uses RNA polymerase? Transcription. Good. Mimics of nucleotides and base analogs. And then translation. If you've ever taken erythromycin or tetracycline, this is exactly what you're interfering with. You're interfering with translation. And those are really common antibiotics. Most people have had them at some point in their lifetime. Okay? All right, so we're going to take a look at changes in the DNA sequence. And we talked about this with thymine dimers about mutations. What is the definition of a mutation? Permanent change in the DNA. Good. And there are different kinds. There are substitutions and frame shifts, okay, where you actually insert and delete bases. So we have missense mutations, okay, this is a lot of vocabulary. Missense mutations where you get a single change in the amino acid. Nonsense mutations, these are catastrophic, okay, why? Because it changes a normal codon into a stop codon. So what happens to the protein? Instead of being like this, what is it? Like that, right. Okay, and then we do have silence or what are called same-sense mutations, okay, that has no effect. Because if that mutation happens in the DNA that gives that third codon a little wobble, sometimes we don't even hear we don't even see the mutations in the phenotype. Frame shifts and insertions, you're inserting bases or taking bases out, okay? All right, so here's some word analogies, okay? Okay, so let's pretend like this is a sequence of DNA. We know it's not, because we don't have an S. We don't have an A. Okay. So the cat sat on the mat. Makes sense. Boring, but okay, makes sense. Okay, here's a missense mutation. Now, what is a missense? Causes a single amino acid change. So something could go in here, okay? Some kind of substitution. A nonsense mutation, what happens there? Shortened, right. The cat, okay, that's pretty short. Same sense, can you tell anything here? No, you might change that last codon, remember the wobble hypothesis? So you might change that last one so the amino acid doesn't really change. The frame shifts are the worst, okay? Frame shifts are, well, nonsenses are bad. Frame shifts changes your whole reading frame. So if we take this out, it becomes vec, ats, atto, him, at. Doesn't make any sense. No, okay? Because what did you do? You changed that whole frame shift, or change that whole reading sequence. So if we take this out, this is good, but now it's this, and whatever. And what happens? You totally bypass that stop codon. Okay? So frame shifts can be a real problem. Okay, so there's some practice things to do. Let's give you a real life example of things that you'll see clinically from one mutation. Okay, we go from CTC to CAC. Okay, one change. What happens? Does it change the messenger RNA? Yeah. Does it change the protein?
Yes.
Yes. It goes from glutamate to valine. Okay. what does it do? It changes the charge, okay? It changes the amino acid now has a charge on it. So what the hemoglobin molecules help the red blood cells keep their structure, and what does hemoglobin do again? What does hemoglobin do? Carries oxygen, right? It's going to carry oxygen. And it has to have the right shape to do it, and there's an iron in the center, and it's got all these arms on here, and it carries oxygen. But if it has this mutation, you get abnormal cells. And this is called, if you can see, this is what is sickle cell anemia. Have you ever heard of that? Yeah, that's a horrible, horrible genetic disease where your red blood cells, instead of being like this, and remember the nucleus pops out here, so you get a little divot, they look like sickles. And so what happens? Is this going to travel through a capillary very easily? No, it doesn't. It gets stuck. And when people go into sickle cell crisis, which can be because they get an infection, they have a change in temperature, they worked out too much, whatever starts that crisis, it's a hospitalization with morphine. I mean, it is pretty serious. One mutation. There's another disease that it results from one mutation, like this. Anybody have an idea what it is? Cystic fibrosis. Remember, cystic fibrosis is a change in the chloride channels. Most cystic fibrosis is from a delta 508 mutation. So you've got this sequence in the DNA at the nucleotide 508, there's a mutation. When that happens, the chloride channels change, so they're always open. And what's the result for the patient? Chloride goes out, water follows. So they get a really, really thick mucus, not only in their lungs, but in their gastrointestinal tract. Tons of infections. A lot of the CF patients that you will see, what do they die of? They die of usually pneumonia, right? Because they've got this really thick mucus that microbes love to live in, right? Okay, and it won't be uncommon to run into cystic fibrosis patients that have multi-drug resistant infections like MRSA in their lungs. Why? They've been on antibiotics forever to try and keep those lungs clear. Yeah?
For sickle cell, it does have something to do with malaria, right?
Yeah, it gives, if you're a heterozygote, so if you have, if you're a heterozygote for malaria, like this, so you have one copy, or a heterozygote for sickle cell, you actually have resistance to malaria. So you're never going to see this trait ever disappear. If you have cystic fibrosis and you're a heterozygote, you have resistance to cholera. So as soon as you see some heterozygote advantage, this is always going to be in the human population. So yeah, one mutation can make a huge difference. So any questions about that? Okay. All right, microbes multiply rapidly. They do binary fission. Okay. Now, if the microbe gets a beneficial mutation, it can outcompete very clear. Because how long does it take for these to go through binary fission? 20 minutes. Yeah, it can outcompete. So when you get a rare mutation, sometimes that's the one that survives in the population. Okay, so this is why we get concerned about antibiotic use. Look at this. If you give this dose of antibiotics to this population, the resistant bacteria are going to survive. And in the next few days, what are you going to have? Mostly a resistant population. Okay? And this is what's led to a lot of problems. I think I'm going to put up a Nova special over the break that you can watch about some of the new emerging infectious diseases. It's absolutely terrifying, some of the things that, and we don't have any antibiotics for them. So, yeah. So if you want a scary movie, Friday night, watch the bacteria stuff. Okay. So, yeah, we've got real problems with multi-drug resistance. So that's what MDR, multi-drug resistance. Okay? And it's not only because of use of antibiotics, but these things are little tramps, I'll tell you. Okay? Yeah. Okay, why? Because okay, they can exchange genetic information 3 different ways. Okay? Humans, we got one way, really. But the bacteria, they go for it. They absolutely just like, they're transferring genetic information all the time. Okay? All right? So you get antibiotic resistance with conjugation and transformation, not usually transduction. Okay? Which ones have we talked about so far? Transformation. And who was the scientist that came up with transformation? Oh boy. Okay, make sure you know that. Who came up with it? Okay. All right. So, in order to understand how these microbes, I mean, we know they do binary fission, that's wonderful, but we have to understand plasmids. And we've seen plasmids before, okay? So plasmids, so here's the bacterial chromosome. The plasmid is usually smaller. It's round, but boy, they carry metabolism genes, like there's a bunch of Pseudomonas that can clean up fuel spills. I have a friend that's got a patent on one that can digest like old tires. So yeah, because you've got a lot of rubber, latex, and that. And he kept breeding the bacteria with these plasmids until it could digest it. So, you know, they can do anything, really. Plasmids can enhance pathogenicity. E. coli, okay, strains that have a plasmid can cause hemolytic uremia syndrome, okay, and we see this with E. coli 0157H7. And then we have methicillin-resistant genes, okay, like this. So, yeah, so those plasmids carry amazing things. And so the bacteria are going to, you know, survive better if they have some of these plasmids, okay? All right, so let's talk about how this works. The first one we're going to see is conjugation, okay? And with conjugation, you want to look at F plasmids, okay? The original name came from like fertility, but we just call them F plasmids, okay? So we've seen this structure before, right? Back in chapter 4, it makes a pili, okay? Goes into here. And when you do conjugation, you have to have an F plus and you have to have an F minus bacteria, okay? Now, what is the F plus going to do? It's going to copy that plasmid and then roll it on into the F minus. So now what happens? Both of these are considered F plus. Okay? So for conjugation, remember the transfer of F plasmids. Okay? And Lederberg is the one that got the Nobel Prize for it. Okay? Okay. So are we good so far? Conjugation. Okay. Now, here's another type of conjugation. Okay, so if we go into, here, we're stuck up here for a little bit talking about conjugation. Okay, so we've done F plasmids. Let's do HFR conjugation. Okay, HFR conjugation happens can transfer chromosomal genes along with the F factor. Okay, so now we're going to get part of the chromosome as well as the F factor. Okay, so here's our F factor that's actually been integrated into the chromosome. It's not called an F plus cell. It's only called an F plus cell if the F factor is if the small plasmid is by itself. It hasn't integrated. This one is integrated, so we got to change the name. What is it called? HFR. What does HFR stand for? High frequency recombination. Okay, high frequency recombination, and that's the HFR. Okay, so what's going to happen when it makes the pili here? This is going to copy, and part of the F factor will be crossing over into this cell, but also carrying some host chromosome genes. Now, what's on those host chromosome genes? Could be just about anything. Could be resistant factors, could be factors that will alter metabolism, we don't know, but it does do that. Okay, so it may get all the way in there, it may not, but it's coming with the F factor plus whatever was in this host. Okay, and so, and guess what it does? It integrates back into the host chromosome. Isn't that sneaky? See why I see they're a little trampy? They just change genes with anybody. Kind of crazy. Any question about HFR? What does it stand for? High frequency recombination. How is it different from the F factor, F plus F minus system? Okay, it's integrated. That F factor is in that host chromosome. Okay, good. Okay, next. Okay, so when you come think about conjugation here, you can list a couple of types of conjugation, right? We talked about a couple of types. What are the types of conjugation? F plasmid and HFR. Good. All right. Okay, let's talk about transformation. We've already seen this before. That has happened here. We saw this when we talked about the experiments that led to, is DNA the genetic material? And what happens is some of these donor DNA fragments, here's the host chromosome, get into the cell, and they're usually single-stranded. And there's a couple of things that can happen. They could just degrade, but it's more exciting for the bacteria if it actually integrates as part of the host chromosome. Okay, we saw that. What kind of bacteria did we use, did we see with transformation back a couple weeks ago? What did we see? Remember we had two phenotypes. What were the two phenotypes? It was R and S, remember that? Okay, yeah, so that's transformation. Oh yeah, here it is, Griffith's experiments. Remember this? Yeah, so, okay. Now, if we go back to our list up here, our thing, so here's transformation. You've got a lot of information to put in here about it, right? And who demonstrated transformation first? Griffith, yeah, okay. So let's go back to transduction, okay? Transduction involves bacteriophages. And what are bacteriophages? They're viruses that infect bacteria. And what do we call one that looks like this? T-even, yeah, wow, you're on fire. Awesome. Okay, yeah. And remember, they're all competing. Here's a virus, and we talked about how, and about lytic and lysogenic cycle, okay? But this is generalized transduction, okay? So a generalized transduction is a plain old vanilla, bacteriophage infection. And it can be, go from lytic to lysogenic. So it can either decide to make lots of viruses and make viruses, or it can stably integrate into the host chromosome. Those are the two choices, okay? All right, so that's generalized transduction, okay? Let's see, what do we got now? This is specialized transduction, okay? This is kind of, we've seen this before, but we didn't label it as such, okay? But when we do, we look at specialized transduction, we have this section in here called a prophage. Do you remember this when we did lytic cycle? Okay, so, and lysogenic cycle. So what will happen is that the prophage, which is some of the incorporated DNA from the viruses, goes and breaks off, makes a little plasmid, and this can start replicating. The phage can replicate, and then it can go and introduce itself into the host bacterial chromosome right there. Okay? So, yeah. So what happens? Well, the products of this is recombinant. This is a recombinant. So let me go back to a couple of things and ask you some questions. Okay? All right. Okay. Is this a recombinant? No. Why? Okay, it's not in the host chromosome. Okay? Is this a recombinant? Yes. Okay. Is this a recombinant?
Yes.
Yep. Okay, yeah. And so, is this up until here a recombinant? No. Okay. But is this down here a recombinant? Okay, good. Okay, so good. Yeah, they interact. Yeah. Do you know how many new antibiotics we've had on the market in the last 5 years? 2. Okay. Are we going to see any more than that for a while? Probably not. It's not economically viable for companies to spend $20 billion to get something on a market when you have 12 infections a year. Okay? That's the state we're in. And it's getting worse. I mean, really. Because the market's getting worse, right? So what happens? So if you look at these, you can see all of these interact where all the arrows go, back and forth. Look at this, Salmonella and E. coli. They share genes all the time. That's why I call them little tramps. Okay, is because they're going to share genes, and you can't just, you're going to take micro right now, but in 7 years, you're going to have a different composition in the field, okay, of these microbes. Okay, there's one we're following in Russia right now that's causing this horrible hemorrhagic, response, and we suspect that it's a bacterial infection, but we're not quite sure. So what could have happened? Well, if you start seeing, hemorrhagic things, I start thinking about E. coli and, all those genes that cause blood cells to lyse. Did they pass them? And how do we know? And, where did this happen? Did it happen in a facility like a SNF? Did it happen in a hospital? Is it remote? What city did it happen in? Is it Moscow or is it somewhere in Siberia? Because you've got to look at what does your population have? You probably noticed this. Your kids, when they go to a new school, what happens? They get sick. They get viral things, but they also get exposed to new bacteria. Are those bacteria now exchanging genes? Absolutely. Yep. So, yeah, it means one thing: job security for you. Okay. All right. Yeah, infectious disease. Yeah, I was talking to one of my docs while we were deployed, and he's like, yeah, I switched from oncology to infectious disease because we're always going to have infectious diseases. I was like, okay. He's like, oh, I have to think about job security. I'm like, okay, kidneys, very good. All right, so we're going to talk a little bit about gene expression, okay? And there's a few things to pay attention to: gene expression in prokaryotes. Now, I asked you to read and get prepped for this, okay? So we're going to turn on the lights and you're going to talk to your neighbor because we're going to talk about something called the lac operon. So without looking at your notes, we're back to the other partner. Please draw the lac operon and explain how it works. Yeah.
The gene specifically, I don't remember, but it encodes for an enzyme that digests lactose. That's the only part. So when lactose is present, it I'm trying to find, like, if I'm remembering correctly, it binds here, and it allows it for RNA to bind to the promoter and encode for the gene, so then it can break down lactose. And once all the lactose is used up, there's nothing here to bind anymore, so that it can inhibitor gets suppressed.
You were in there. You remember this?
I think that's how it went.
Okay. How'd you do? Where's your lap? Oh,
I just like
okay.
it's been a while, I admit, since I read this chapter, so
Okay.
I didn't refresh as I should have.
That's okay, we'll get it right now.
Yeah.
Okay, let's answer some things about the lactoperon that should maybe have come to mind. What is it? What is the whole purpose of the lactoperon?
Gene expression.
Yeah, it's making genes for what? It makes genes to. It makes protein. It transcribes the genes to make the proteins to digest what?
Lactose.
Lactose. What is its normal state? Off. Right, off. Okay? So is it an inducible or a repressible operon?
Inducible.
It's inducible. It's going to be there and then you can turn it on. Okay? All right. How many genes are associated with the lactoperon? 3. Good. Okay? So let's take a look at how this all works. Okay? Yes, I know it might be harmful. Come on. Okay. There's a little video embedded in here, but in the interest of time, we'll kind of skip it, but you can go through and do it. If you look at the operon, you've got a promoter, an operator, lac Z, Y, and A, which are the genes, right? Okay? All right, one is for beta-galactosidase, one is for inputting that, and the other one is for cleaving that bond that lactose is, okay? So, if it's off, what's going to happen to the repressor? The repressor is going to be there. The only way it can be removed is how? The lactose binds there and pulls it off. Now, if you've got steric hindrance, that's what this is called, that move, so the polymerase binds and there's a big repressor there. Can these genes be transcribed? No. Okay, so it has to be pulled off. And if there's lactose in the environment, it will bind to the repressor and pull it off. Isn't that clever? That's really clever how it does that. Now let me ask you something. Is this DNA or RNA?
DNA.
Is it DNA or RNA? Wow, that's a variety of questions. You have 2 choices. Okay, here's a hint. What is here? RNA polymerase. So what are you going to be making? So what does this have to be? DNA. Yeah. Yeah, look for cues. Look for cues. Yeah. Lactose intolerant people, well, first of all, we don't have a lactoperon. Okay, this is in bacteria. Okay, so in lactose intolerant people, they just don't make the enzyme they don't make lactase. So if they can't make that enzyme, they can't break down the sugar is very irritating and, get all the symptoms. Okay, all right, so, I'll have you draw that. Okay, let's do the lactoperon. Can you draw it? Oh, I'll turn on the lights in between runs. From memory, don't forget. Yeah, memory, memory. Where did it come?
No, but anyways, underrated. I think it is gonna be this one. If you look at the thing, we have every letter except I. We have promoted. No, we don't even have O. It's EYA. Or does it go? No, because EYA is the
Okay. Do you think there's going to be a section on this on the exam? Yes. Okay. I put lots of videos, lots of ways. You know this. You got to know this part of gene regulation. Okay. Now, there are repressible operons. Okay. Okay. Operons are mostly on. Okay. Like tryptophan. What is tryptophan? What kind of molecule is tryptophan? It's an amino acid. It's one of the cutest ones. If you don't know what tryptophan looks like, take a look at it. It is adorable. Okay? Tryptophan always has to be on because that's an amino acid that is made. So it operates a little differently. Okay? So you've got the promoter, you've got the operator start here. Okay? You've got a repressor that comes in when you've got enough tryptophan in your system, or the bacteria do. Have enough tryptophan that they can shut down for a little while. But these are always on. Arginine, always on. Okay? So they're a little bit different. I don't think I put a video on with that. I'll try and find one that is decent, that talks about the tryptophan and arginine operon. Okay? But these are repressible. Why? Because they're always on. Okay? So do you get the difference between inducible and repressible? Okay, all right, cool. So, here we go. Yeah, and this is actually feedback inhibition. Once we get enough tryptophan in that, you can put the repressor will bind here, and then the promoter, the RNA polymerase will be stuck on the promoter. So on the promoter, you've actually got an operator site there that this can bind to. Okay, so that's pretty cool. All right, so lots of words here. Yeah, it's all regulation. We can do, proteins can help shut off genes. We just saw that. When sugars can help turn on genes. Okay, and it's constantly changing. So that's the end of chapter 9. On Thursday, we've got one more to do, right? Oh, Wednesday, yeah. What day is this? This is Monday. Okay, we got some time. So please look at chapter 8. Now, before we go, I have a question to ask because this came up in the night class. Yo, I got you for 2 minutes. All right. Those of you who have an electronic version of chapter 12, are there images where you get an error message where it said it can't be shown? What? Oh, edition 12, the electronic edition. If you bought the electronic edition of this book, the latest, which is 12, are there any images that do not come up that gives you an error message? Okay, I want to see an email from you with a screenshot of that. Anybody else that's not coming up? You get all your images. You have the 9th electronic division or edition? My brain is going so fast. I need to slow down. Okay, how did you get the 9th? Did you buy a 9?
Yeah.
Okay. All right. So it's just a 12. I need a screenshot of that. Okay. All right. Anything that comes up on your electronic editions that says there's an error, I need to know about it right away. We had a meeting about it on Friday, so I want it, we're gonna try and work it out so you can see it, okay? All right, if you did pick up your Quizzy Wizzies, they're up here.